Okay, so this will be my video going over skeletal muscle cells and their excitation-contraction coupling phase and the relaxation phase. Okay, to start off, we have a somatic motor nerve, and in this somatic nerve, we will have a bundle of axons. And when a signal is sent down these axons, it's the action potential, and it goes all the way down until it hits the end of the axon at the axon terminal. In neurons that are connecting to skeletal muscle fiber cells, this is called a neuromuscular junction. Neuro for neuron, muscular for skeletal muscle, and it's a junction. It's meant to connect to a muscle fiber cell. So at this neuromuscular junction, when the action potential arrives, it will trigger voltage-gated calcium channels to open up, and calcium starts flooding in. Calcium flooding in will trigger these synaptic vesicles to undock, to release their neurotransmitters that they have inside. In neuromuscular junctions, the neurotransmitter will be acetylcholine. Okay, so acetylcholine is released at the synaptic cleft, and it will arrive at the motor end plate of the muscle cell. The motor end plate is just like the point where the junction where they meet, right? So in the motor end plate, we can see that there are these invaginations of the sarcolemma. They're not transverse tubules. Their only purpose is to maximize the surface area of the sarcolemma so you could fit more receptors and all that stuff. So the more receptors you have, the more acetylcholine you can accept, and the more depolarization that can happen with the muscle cell. Okay, so we see here at the motor end plate that there is a specific receptor protein called the nicotinic receptor. Acetylcholine is the key to this lock, to this nicotinic receptor. When acetylcholine binds, unlocks the door, if you want to think of it like that, sodium comes flooding in. What you don't see depicted here is that potassium is also present, and when it's stimulated to open, it will also go flooding out, because we know that potassium is high inside, low outside. I don't have it drawn out here. Our PowerPoint doesn't have it drawn out, and I think the textbook doesn't really have it drawn out, because the emphasis is put on the depolarization that sodium is doing to the inside of the cell, not really what potassium is doing when it leaves. So we just want to have that emphasis placed on sodium. Still need to know that potassium is doing a thing, or it's present, it's leaving. But sodium is our focus. So sodium flooding in will cause a depolarization of the cell and of the sarcolemma. We can see that it's traveling down the sarcolemma until it hits a T-tubule, a transverse tubule, and it travels all the way down. In these transverse tubules, we have a protein here, and this protein is called a dihydropyridine receptor protein. And note that I did not say it's a channel. It is not a channel. It's not letting anything come out or in. It is a receptor protein. What it does do is it opens, I like to think of it like a door. So when there's a change of voltage down the sarcolemma of the T-tubule, an action potential, when it hits this DHP, it tells its arm, this strand of protein, it tells it to perform a conformational change. I like to think that it has a little hand right here, and when it's being told to move, it will open this door. And what door is it opening? It's opening a ryanodine receptor channel. And this ryanodine receptor channel is located on the sarcoplasmic reticulum of the muscle cell. The sarcoplasmic reticulum is essentially a storage depot for calcium, right? We have several parts to it. We have longitudinal tubules, and then we have the terminal cisternae, right? Terminal cisternae are going to be located really close to the DHP because the terminal cisternae have these ryanodine channels. So when the action potential travels all the way down, tells the DHP to open the door, it will use its arm, open the gate, and boom, we have a super high concentration of calcium out here, low concentration out here, so high to low is the flow, so it starts flooding out. Now, we can see that we have our sarcomeres really, really close by. So, calcium stimulus will come and bind with troponin. When it binds with troponin, it tells this tropomyosin strand to move off of the actin binding sites. When that occurs, if everything else is going to plan, the myosin head will cock up and perform the power stroke, which essentially moves this thin filament towards the M line, will cause it to slide over to the M line, which is what a contraction is. So that is the excitation-contraction coupling phase. How do we go into relaxation? Well, the first step is for the brain or whatever to stop sending impulses, to stop sending action potentials. We don't want to move anymore. Okay, boom. Acetylcholine's not being released. Nicotinic receptors no longer being triggered to open. Sodium's not flooding in anymore. No more depolarizing stimulus inside of the cell, which also means no more action potential traveling down the sarcolemma, down the transverse tubule. Okay, cool. But we still have a problem. All of these calcium stimulus that are still out here are also still, they're still present. They're just hanging around. They have nothing to do, right? How do we remove the calcium stimulus? Because if we don't remove the calcium stimulus, our sarcomeres are going to continue contracting. The movement's going to continue going, even if we don't want the movement, even if our brain's not telling, like, hey, I'm not sending action potentials anymore, like, stop moving. So what do? So we have this primary active transport located on the longitudinal tubules of the sarcoplasmic reticulum. These ones are called calcium ATPases, or calcium ATPase pumps. So what happens, since it's primary active transport, it will hydrolyze an ATP, and it will kind of vacuum up all of the calcium stimulus that are still floating around to recycle it. We want to reuse it. So calcium is being sucked right back into the sarcoplasmic reticulum. The lowering levels of, calcium in the sarcomeres, which will then mean that troponin is no longer being stimulated. If troponin is no longer being stimulated and ATP is present, ATP will bind to the myosin head, causing it to release. And since troponin is no longer being stimulated, it will move tropomyosin back into its resting position of covering the actin cells. the actin binding sites of these globular actin monomers. And that will stop contraction of sarcomeres, which will then stop skeletal muscle from contracting. So those are our 2 stages. And if we go a little closer look into, or if we do a little closer look into like the myosin head activity, we see that we have a couple of, phases. I like to note though that this isn't like step 1, step 2, step 3, like very rigid formation. It's very fluid. As one thing's happening, the other thing's already beginning to happen. So it's like a dance. It's happening very fluid-like. So we could start off from, ATP binding, calcium stimulus is leaving. We are in our relaxed state. The only way that these myosin ATPases can relax, can release this, is if ATP is present. If ATP is not present, it will stay stuck essentially to these actin binding sites. So ATP binds to the myosin head, causing it to release from the actin binding site. Calcium stimulus is gone, so troponin moves tropomyosin back onto the sites to cover them again until calcium stimulus is introduced. So that's when we go into our cocking phase. When stimulated, it will bind to troponin. Tropomyosin will move off of the site. The ATP that was here will be hydrolyzed and turn into ADP plus 1 inorganic phosphate. It being hydrolyzed causes it to store the potential energy into this bond right here. So we have like a rubber band being pulled really taut. You still haven't released it, but when you do release it, it's going to go flying, but it hasn't released. It's just being stored right here. So we have the energy stored right here. ATP is gone. It's now ADP plus 1 inorganic phosphate. And then, with calcium still being present, ADP will release that inorganic phosphate and release the potential energy that's stored into this bond, performing the power stroke. And the myosin will move the actin filament, the thin filament, towards the M line to cause overlap. Then, shortly after, myosin will release the ADP that it had on its head. It will release it, and it will stay bound here, even if calcium is leaving, because it needs an ATP to release. It's kind of like glued on. You need the ATP to make the myosin head release. Now, ATP is present, causing it to release. Troponin is not being stimulated because calcium is gone, and that puts it into relaxed state. Okay, so that makes up skeletal muscle, and let me know if you have any questions.